Very good afternoon. Today we are going to discuss heredity and what evolution related audio notes. Okay, for SSLC and 10th standard students. Okay, we already making like these several videos. These videos are helpful to as audio notes at the time of exam, to make a revision, to make a notes, or to read or to write affairs. Okay, these videos are helpful. That's why I'm preparing these videos. So let's start. Okay, first of all, what do you mean by heredity? Okay, the process of transfer of characters or traits from the uh, parents to their offsprings, it's called as a heredity. Then what do you mean by variations? The differences between the characters or traits among the individuals of the same species are called variations. Okay, next, if we see the same parental kids, some are different in color. Okay, in sparrow also and termites also. Next, for example, human height is a trait which shows different variations like tall, short, medium. Another example, earlobe. It shows two variations: free earlobe and attached earlobe. Then the variation is necessary for evolution. The great advantage of variation is that it increases the chance of survival in a changing environment. Then next, accumulation of variations during a reproduction. When organisms reproduce, the offspring show minor variations due to inaccuracies in DNA copying. These variations are less in asexual reproduction and more in sexual reproduction. Some variations are useful variations and they help the organism to adjust to the changes in environment. Some variations do not help the organism to adjust to the changes in environment and they may be die and become extinct. Okay. Then see they given here variations. Characters are transferred through DNA molecules present in the gene on the chromosomes which are present in the nucleus of the cell. The inheritance of characters is due to the fact that both the father and mother contributes equal amount of genetic material to the kid or child. So for each trait there are two factors, one more from the father and one from the mother. We already studied a cell. In that cell a nucleus is there. Okay, in that nucleus quite like structured genetic material is there. In that genetic material we observe what chromosomes. These chromosomes, structure of a chromosome, this is a, okay, in human beings 23 pair of chromosomes are there. Whenever we again, okay, divide the chromatids, this is centromer and these are chromatids. Again, whenever we divide chromatids, we get a coil like structure. In that coil like structure, nitrogen bases are there, pentose sugar is there, phosphate is there. At last again, whenever we make a what a simpler and simpler, we get a what, a DNA structure. What we get? DNA structure. DNA is a what, very important structure in our body. It has genes, thousands together genes. Each gene is what, derives our characters. Then next, chromosome. Okay, the structure of chromosome they give are here. This is already explained for you. A gene is a unit of a DNA on a chromosome which transfer characteristics from parents to their offsprings during a reproduction. Then a gene is the unit of inheritance or heredity. Next, genes for controlling the same character an organism can be of two types: dominant and recessive genes are there. Dominant and recessive. Then what do you mean by dominant? The gene which decides the appearance of an organism even in the presence of an alternative gene is called as a dominant gene. It is called as a whatever the genes what appear more and more in their progeny, that's called as a dominant gene. And whatever the gene, okay, it's an inactive stage or what, it appears very less in the next progeny or next offsprings, that's called as a recessive gene. The gene which can decide the appearance of an organism only in the presence of another identical gene, it is called as a recessive gene. Then next, the dominant gene is represented by capital letter, corresponding recessive gene is represented by small letter. For example, dominant gene for tallness is represented as a capital T, and the recessive gene for a dwarfness, it is represented as a small t. Then genotype and phenotype. What do you mean by genotype? The genotype is the description of genes present in an organism. Phenotype, the characteristic or trait which is visible in an organism is called as a phenotype. Example, tall or dwarf. Mendel was the first scientist to make a systematic study of patterns of inheritance, which involved the transfer of the transfer of characters from parents to their offsprings. Okay, father of genetics, Mendel, John Gregor Mendel. Mendel chose a pea plant, Phycum sativum, for studying inheritance due to the following reasons. Several reasons are there, very few reasons they given here. Pea plants have variations for many traits. Tall and dwarf pea plant, green and yellow seed pea plants, round and wrinkled seed plants. Okay, pea plants are self-pollinating also as well as cross-pollinating also. That's why we can able to carry experiments. Pea plants have a short lifespan. Within a year, 3 to 4 experiments we can able to carry. Then they can be grown easily. Where in small ponds also, or small pots also, we can able to grow them easily. Then first generation, when two parents cross to produce their offsprings, then the offsprings are called first generation or first filial generation. Then second generation, F2. When the first generation plants cross among themselves to produce offsprings, then the offsprings are called F2 generation or second filial generation. When the plants having a one pair of characters, example tall and short plant, was crossed, then it is called as a monohybrid cross. It is called as a monohybrid cross. Ekatalikarana in Kannada. Mendel selected a pea plant having a one pair of characters, a tall pea plant and a short pea plant. He selected pure tall and pure short, capital T and small t pea plants and cross-pollinated them. Okay. Uh, he obtained all tall plants, capital T small t, in the first filial generation. When the first filial generation plants were self-pollinated, he obtained a tall and dwarf plants in the ratio of 3:1 in the second generation. The ratio of pure tall plants, capital T 1 is there. Then hybrid tall means capital T small t 2 are there. Then pure dwarf small t 1 is there. Then what we can able to say, their ratio is 1:2:1. Then next, the trait that expressed the trait that expressed the trait that is expressed in the F1 generation is called the dominant trait, and the trait that is suppressed in the first filial generation, it is called as a recessive trait. They took here three examples based on tall and short plant, based on a color of flower, based on seeds what structure, round and wrinkled. They explained here. See, in first generation, Mendel, what he done in pea plant, short and tall plant he crossed, okay, by cross pollination. Then in first filial generation he got all four tall plants, okay, their traits are capital T small t. Then in next generation self pollinating, he got what F2 generation. In F2 generation, what, three tall plants and one dwarf plant. In these three, one pure is there and two impure is there, and one, but another one, whatever the is there, that's a impure, purely impure. Okay. Then next, okay, purple color flower and white color flower classed in first 5 year generation cross pollination, he got the all purple flowers, okay, capital P. In next generation, three purple and one white flower, then their ratio is 3:1. Okay, it's a what phenotype ratio. Genotype ratio 1:2, 2:1. phenotype, it's a genotype. Then phenotype 3:1. Then based on seed, in pea plants two types of seeds are there, round and wrinkle seeds. Okay, he cross pollinated and he got all round seed plants in first filial generation. Okay, all round seeds. Okay, their traits are capital R, small r. Then again he said what took plants from these, what again he took seeds from these plants and self pollinated, and he sowed the seeds. Then in second filial generation he got three round seeded plants and one wrinkled seeded plant. In these three what round seeded plants and one wrinkled seeded plant, it's called as a phenotype. And another one, which one? Okay, one pure round seeded plant, two impure round seeded plants, and one wrinkled seeded plant, it's a 1:2:1, it's a genotype. It's what? Genotype. Next. Okay, same. When plants having a two pair of a characters, example shape and color of a seed, were crossed. When plants having a two pair of a characters, example shape and color of a seed, were crossed, then it is called as a dihybrid cross. Mendel selected a pea plant having a two pair of characters, shape and color of a seed. He selected the plants having round yellow seeds, capital R, capital R, capital Y, and wrinkled green seeds, small r, small y, and cross pollinated them. Then after cross pollinating in first filial generation, what he got? He got all round, all what? Round yellow seeds, but their traits are not pure. Their traits are capital R, small r, capital Y, small y. Okay, in first filial generation. Then again by self pollinating, in self pollinated in second filial generation, out of 16 plants, he got what? 9 round yellow, and then 3 round green, then 3 wrinkled yellow, and 1 wrinkled green. That's why what it's what phenotype is 9:3:1. It's a phenotype. Then genotype 1:2, 1:2, 1:2 is to 1:2 is to 1:2 is to 4, then is to 2:1, is to 2:1, like this. What genotype will come here. Then see, in first filial generation, whatever he took, pure round and yellow seeds and pure wrinkled and green seeds. Okay, in first filial generation, what he got? He got capital R, small r, capital Y, small I, all plants. And what in second filial generation, again he took those seeds and sowed and self pollinated. After self pollinating, he got the ratio 9:3:3:1. I told for you. Then see, whatever the round yellow traits, from these round yellow, round green, then wrinkled yellow and wrinkled green, 4 traits are there. In Punnett checkerboard, he took both sides, this side also and this side also, and cross pollinated. After cross pollinating, he got the result, whatever the results are there. Okay, 9:3:3:1 he got. And he got the genotype, whatever the genotype I explained for you. Okay, see, these are the different boards of a what? Dihybrid cross. Okay, I already explained in Kannada medium, like what monohybrid cross and dihybrid cross for 10th standard separately. I made this video in Kannada medium. If you want to see there. Then sex determination in human beings. Okay, human beings have a 23 pair of chromosomes in the nucleus of the cell. Out of this, 2 chromosomes are sex chromosomes, X and Y. The female has 2 XX chromosomes. Okay, and a male has 1 X and 1 Y chromosome. The sperms and eggs have one set of sex chromosomes. Some sperms have a X chromosomes and some have a Y chromosomes. All eggs have a X chromosomes only. If a sperm having a X chromosome fuses with an egg having a X chromosome, the child will be what? Definitely girl. Then if a sperm having a Y chromosome fuses with an egg having a X chromosome, the child will be boy. That's why sex determination is not depends on females, it depends on males, but what in males it's not in hand. Artificially they can't produce male and female if they are male, but they can't produce because it's a natural process. The movement of a chromosome X or Y we cannot identify. That's why in males what type of chromosomes are there? XY. In females what type of chromosomes are there? XX. Then whatever the chromosome in males are there, okay, males release the sperms. In these sperms X chromosome is there, means what from females definitely X chromosome, always X fuses with X means what, the newborn baby is female. If a sperm contains a Y chromosome, it fuses with the female X chromosome, then the newborn baby will be male. Then variation may or may not help organism to survive. Some variations help organism to survive. Example, there are some beetles living in green bushes. They increase their numbers by reproduction. Crows can easily see the red beetles and they are eaten by the crows. During reproduction, due to some variation, it may be DNA replicating variation or any other reason, variation is caused. That variation, some green beetles are produced instead of red beetles. The green beetles are not visible to crows and are not eaten by them. Then gradually the population of the red beetles decreases and the population of the green beetles increases. This variation has helped the organism to survive. Next B. Some variations do not help organism to survive. During a sexual reproduction, a color variation occurs in red beetles, and what they are turned into what, some blue beetles are produced instead of red beetles. Both the red and blue beetles are visible to crows and they are eaten by them. Then the population of both red and blue beetles decreases. This variation has not helped the organism to survive. The next question. Acquired traits cannot be passed from one generation to next. If the population of a beetle increases and the plants are affected by disease or what flood, drought, any of the reason, the food is not available for beetles, then what the beetles what decreases and their body weight also decreases. If after a few years the availability of food increases, then the body weight of the beetles also increases. This acquired trait cannot be passed from one generation to next generation because there is no change in the genetic composition. Whenever the change in the genetic composition, it may be in DNA, then only it carried to next generation. Then it was only heredity, otherwise it's not heredity. Then speciation. Okay, formation of a new species from existing species. Speciation, formation of a new species from existing species, it's called as a speciation. The formation of new species from existing species is mainly due to one or more of the following factors. They are accumulation of variation, physical barriers, genetic drift, natural selection, and migration. Accumulation of variations, the difference between the individuals of the same species is called variations. The accumulation of variations over several generations produce a new species. Then physical barriers. Population may get separated by physical barriers like mountains, rivers, lakes, etc. These isolated groups produce variations which can produce new species. The next genetic drift. Natural calamities or introduction of a new members of the same species in an area can produce changes in the gene pool of the population and new variations are produced which can be produced new species. The natural selection. Only those individuals of a species which have useful variations and can adapt to the changes in the environment survive and the others die. These organisms can produce variations and new species. The next, migration. Some individuals of a species may migrate to new geographical area and adjust to the changes in the environment there and develop a new variations and produce a new species. Okay, this is also causes for speciation. Then now we are going to discuss what evolution. A gradual changes takes place in living organism, giving rise to new organism due to change in their genetic composition is called as a evolution. Okay, monkey to man, it's example for evolution. Then inherited traits. Inherited traits are traits in an organism due to changes in the genetic composition, and it can be passed from one generation to next, and it results in evolution. Then B, acquired traits. Acquired traits are traits which are acquired by an organism during its lifetime, and it cannot be passed from one generation to the next, and it does not result in evolution. Then evidence of evolution. There are number of common features in different organisms which provide evidence to show evolution relationship. The main evidences of evolutions are from the study of homologous organs and analogous organs and fossils. Then what do you mean by homologous organs? What homologous organs are organs which are similar in structure but are different in functions. The forelimbs of amphibians, reptiles, birds, and mammals have a similar structure but different functions. Different function. Frog, it's an amphibian, uses its forelimbs to raise the front of the body. Lizard reptiles uses its forelimbs for the walking and running. Bird, what forelimbs are modified as a wings for flying. Mammals use forelimbs to what forelimbs for a grasping, walking, running, swimming, flying, etc. This shows what evolutionary relationship. Structurally, they are similar, but their work they are doing different works. Okay. Example: human, whale, dog, bat. Different selection. What selection? What pressure? Then analogous organs. Analogous organs are organs which are different in structure but similar in function. Example: wings of a butterfly, bird, and bat have different structures, but they are performing similar functions. This shows evolutionary relationship. What kind of evolutionary relationship? Analogous organ. Okay, bat's wing, bird's wing, and insect's wing. They are not similar, but they perform a similar function. Then homology and analogy. Homology and analogy. Okay, homology, it's about homologous organs. Analogy, analogous organs. Structurally they are same, but they are doing a different function. Which one? Homology. Analogy, structure is different, but they are performing a similar function. Fossils. Fossils are the remains of organism which lived long ago. From the study of fossils, we can know their structures and the time period in which they lived. The fossils of complex and recent organisms are found closer to the surface of the earth, and the fossils of simpler organisms are found deeper inside the earth. The age of fossils can be determined by radiocarbon dating. The study of fossils shows the evolution of simpler forms into complex forms and their evolutionary relationship. Okay, tree trunk fossils, fish fossils, it's a with Cnitia. Then limonite, different fossils they given here structure. Evolution by stage. Complex organisms and its organs developed from simple organisms gradually cover generations. Evolution of eye. The eye of planaria are just eye spots to what detect light. It developed gradually into complex organ in higher animals. Evolution of a feather. Feather were the first developed in dinosaurs and used for protection from cold. Later, birds used them for flying. Then, what evolution by artificial selection? Humans cultivated a wild cabbage for over 2000 years and produced different vegetables from it by artificial selection. Example: cabbage by selecting short distance between leaves, cauliflower by selecting sterile flowers, kale by selecting large leaves. Okay, then kohlrabi by selecting the swollen stem, broccoli by what swelling flowers growth. To study evolution, embryology also helps. Here they given different animals embryo. Example, here lizard embryo is there, tortoise embryo is there, pig's embryo is there, human beings embryo is there. All four structure is same. But whenever they are growing, see, slight structure is changed. And whenever they come to at that what we call mm, what we call at the time of pregnancy or what delivery, their structure changes. They develop different legs and hands. Okay, this also helps to study the evolution, how evolution occurred on earth, what are the similarities in between us and other organisms are there. We all evolved from a same organism. Okay, which evidence shows embryology, because all embryo are same. Then evolution should not be equated with progress. Evolution has not resulted in progress. Evolution has resulted in formation of several complex species from similar species due to variation, genetic drift, and natural selection. This does not mean that one species get eliminated and when a new species are formed, or that the new species are better than the older species. It's not like that. Species get eliminated only if they are not able to adapt to the changes in environment. Several species which could not adapt to the changes in an environment still continue to survive. For example, bacteria. Human beings have not evolved from chimpanzees. They had a common ancestor from which they evolved separately. Human beings are not the what pinnacle of evolution, but they are only the what species among the several evolving species. Human evolution, Homo sapiens. There is a great diversity among human beings in their form and what feature around the world. Human beings evolved in Africa, some of them stayed in there, and others migrated to different parts of the world. Then, due to genetic variations and in a developmental changes in different geographical regions, what they developed changes in their forms and feathers. Okay, then see first Lucy. Okay, different human being similarities, but monkeys, apes, and chimpanzees. Okay, okay, up to now we discussed what evolution and heredity chapter related audio notes. Hopefully these audio notes are helpful for your exam preparation, to make notes or to write a fair books, and to very important to make a recall or revision. I told for you several times, whatever the videos I prepare, these are not live classes. In live classes, monohybrid class and dihybrid class, how you want like that I explain. But here you will get only notes. Okay, these videos are helpful to those people, those what they are doing their daily works, because some kids they have their agriculture work, or what some kids they go to what some other works like a paper distributing, or some people what kids they travel for 20 to 30 kilometers to their schools. At that time what they what waste their time. To avoid this, whenever you are traveling or you are doing other works, listening music is not worthy for you. That's why to hear these audio notes, for some people, not for all people. Okay, whatever they can utilize this, not misuse this. Okay, whatever the people they take a use of this. Okay, for them I prepared these videos to recall or to make a revision. Okay, hopefully useful, then take use, otherwise avoid it. Thank you so much.